Hello, dear students. So today I am going to talk about the uh, first topic of the fourth unit of molecular biology and genetic engineering, which is the polymerase chain reaction. So that is also called the PCR, and you have done PCR in your practicals in the genetics as well as in molecular biology. So it is basically the amplification of the DNA in vitro. In vivo, we call it as replication, whereas in vitro, if you want to amplify a copy of DNA, it is called PCR or polymerase chain reaction. And the most common thing we use is a enzyme called the tag DNA polymerase. Also, you require the primers because we all know that the DNA polymerase cannot synthesize the strand de novo, but it requires a free 3' OH group, which is provided by the primers. So primer will also aid in setting the target sequence to be amplified. So flanking primers, the 5' and the 3' end will give you the sequence in between which has to be amplified. So the reaction is then repeatedly cycled through a series of temperature changes. As you have seen in the practical, we use higher temperature for denaturation, and then subsequently we lower down the temperature for annealing of the primers, and then subsequently we use an extension step at the 72 degree where the DNA polymerase extends the length of the primer and based on the complementary base pairs, and it has got several applications both in research as well as in diagnostics. Most commonly, the COVID-19 detection is done by the qPCR. I'll get to know what exactly is a qPCR. Also, in the forensic analysis of DNA, routine DNA cloning, we use the PCR-based methods. So now let us see the history. So Carey B. Mullis was the inventor of the PCR technique in 83 when he was working as a chemist in the Cetus Corporation in California. And for this, he got the Nobel Prize 10 years later in 93 in chemistry for his discovery. During that time, he was using the normal E. coli DNA polymerase. And what he used to do was, after the denaturation step, he used to add the DNA polymerase, and then the extension will occur. And then, after the second round of denaturation, he will again add the E. coli DNA polymerase. So he had to add multiple times that DNA polymerase. So then he thought, why can't he use such a thermostable DNA polymerase, which is stable at even at the higher temperature. So then he isolated the polymerase from the Thermophilus aquaticus, which is a bacteria, Thermophilus bacteria, which lives in the hot springs. So, and its temperature optimized around 70 to 72 degrees Celsius. And that was the best to be used, and you then need to add it only once rather than multiple times. Now let us see the components of PCR. The very first thing is the tag DNA polymerase, which was isolated from Thermophilus aquaticus and lives in the hot springs, and its temperature optimized around 70 degrees Celsius. Now other DNA polymerases have also been used, like the TFL isolated from Thermus flavus, TFU from Pyrococcus furiosus, TLI from Thermococcus litoralis, and TGO from Thermococcus gorgonarius. And all these have been isolated from the thermophilic bacteria. So if you see the different conditions, the PCR optima, extension time per kilo base means every minute it adds around 1 kb, 1 kb, then 2 kb. That means it is more efficient. 2 kb and 2 kb. Then extension temperature is more or less same for all of them. Then magnesium concentration, it requires 1 to 4, 1 to 4, and here it requires 2 to 4, 2 to 4, 2 to 4. pH at the 25 degrees Celsius, it needs 7 to 7.5, except for the PFU, which needs 8 to 9, all others need the 7 to 7.5 pH range. Then DNTP concentration is 40 to 200 millimoles. For TGO, it is just 200. Primer concentration is same for all. Then comes the 5' to 3' exonuclease activity. So 5' to 3' exonuclease activity means the strand displacement activity. So if you have some strand already there and you have the DNA polymerase extending, so it will displace that strand and synthesize, continuously synthesizing its own complementary strand, which it was doing. Also, so this activity is found in the tag DNA polymerase TFL, but not in rest of them. Whereas 3' to 5' exonuclease activity, that means when the polymerase goes back and cross checks whether it is incorporating the correct nucleotide or not. So that means the proofreading activity. So proofreading activity is not there in tag, not there in TFL, but in rest of them. So for the proofreading activity, so such enzymes can be used, like the PFU is mainly used for the cloning experiments and all, when you want the sequence to be correct. And the error rates is 5 into 10 to the power 4, 10 to the power minus 5, minus 4, minus 5, minus 6, and minus 6. So the most error rate is by your tag DNA polymerase because it lacks the proofreading activity. And the PCR product ends for the tag and TFL are the 3' A. So it is a tag. What it does is, after the PCR is over, an extra A is added at the 3' overhang. So you have this 5' to 3'. So this is 5' and this is 3'. So here an extra A is added, and this is also 3'. Here also an extra A is added. Okay, so this is what I wanted to tell. So this we all know. Then basic properties of DNA polymerase used in PCR is, the very first thing is thermal stability. That means it should be able to tolerate high temperature and should be retaining its activity. Then the extension rate, that means speed at which it adds up the nucleotide per second per molecule of DNA polymerase. So earlier DNA polymerase used to have the extension rate of 1 kilobase per second, but now with the modified polymerases you can even have 4 times more, that is 4 kilobase per second. Fidelity refers to the frequency of incorrect nucleotide per kilobase. So higher fidelity polymerases would be more accurate or more precise polymerases, which have the ability to incorporate lesser and lesser incorrect mononucleotides. And the last one is processivity. So processivity means the average number of nucleotides the polymerase adds in just one binding event with the template. So high processivity is very important for amplifying the longer DNA fragments. So there are 4 different classes of DNA polymerases. The first one is the standard thermostable DNA polymerase, which is suitable for the routine PCRs like the one you have done in your practical. So it is just for detection of the amplified product and estimation of the product size. So as I have told already that the tag DNA polymerase have the single A overhang at the 3' end. So this would be this kind of DNA PCR product is good for the TA cloning. So the T factor will have the T at the 5' ends, whereas the PCR product has got A at the 3' ends. So then the TA and A can complement and form the whole construct. So the newer tag polymerases have the good processability, fast extension rate, but they don't have the proofreading. That means 3' to 5' exonuclease activity. So that means they cannot be used for amplifying the fragments for the cloning and expression because we need to have a foolproof sequence without any errors for cloning experiments or the expression studies, because we don't want any mutation in them. Also for the mutagenesis studies, we cannot use this one because we only want the desired mutation to occur. We don't want the undesired mutations to occur. Then comes the hot start piece polymerases. So hot start polymerases are the ones which is used for the non-specific amplification. So like the tag DNA polymerase shows its activity even at the room temperature. So even if the tag is at the room temperature, although they optimize at 72, it will continue amplifying non-specifically also. So wherever it finds a free 3' OH, it will bind and it will start amplification. So this generates a lot of non-specific products during the PCR. So what people have done is during with the hot start polymerases, the polymerase enzyme itself is inactivated by either the chemical modification or with the help of an antibody, which makes the polymerase inactive at room temperature, and it gets activated only when the temperature is raised. So here the example shows where you have the polymerase which is bound to the antibody, and when the denaturation temperature is there, then only the antibody, which is a protein, gets denatured and the polymerase is free and can perform its activity. So this is inactive at room temperature, active at the higher temperature. So this actually greatly reduces the non-specific priming and primer-dimer formation. So the chemical hot start can require up to a 10-minute activation step. But for antibody mediated denaturation, you need just 2 minutes of inactivation step. So hot start is useful when you have the DNA template in this lesser amount, and or the DNA is more complex, or there are several pairs of primers being used, that means in the multiplex PCR. So this hot start polymerase also lacks the proofreading activity, so cannot be used for the cloning or expression or mutagenesis studies. So example is hot start tag DNA polymerase. The third type is high fidelity polymerases, which is also called hi-fi. So they are the proofreading. That means it has 3' to 5' exonuclease activity and can remove erroneously attached the bases during the incorporation event. And so this actually increases the fidelity or accuracy of the DNA synthesis. So this kind of polymerase can be used for cloning and expression studies and mutagenesis experiments. So hi-fi polymerases also removes the A overhang which might be there at the 3' end. So the PCR fragments amplified using this kind of polymerase require the blunt end cloning. They can't go for sticky end cloning like the TA cloning, but they need the blunt end cloning. So example is Fusion High Fidelity DNA Polymerase from New England Biolabs. This is a commercial company. Then the polymerase for amplification of long amplicons, that means you need a high processivity enzyme. So that means a proofreading polymerase is required for the long amplicons. So this is achieved by blending the thermostable DNA polymerase with a proofreading enzyme. So with an optimized buffer and high salt concentration, the PCR products from the genomic DNA can be as long as 25 kilobase in size. So the specific process, the long amplicon polymerase is long amp tag DNA polymerase, which is a blend of tag and deep bent DNA polymerase from New England Biolabs. So in conclusion, you should choose your enzyme for general and routine PCR and ordinary thermostable polymerase like that. For gene expression and mutagenesis experiment, use a proofreading enzyme like high fidelity one. For cleaner product and high yield, you use hot start polymerase. And for long amplicon, use a long reach polymerase. Now, the next important component is the primers. So, polymerase can only make DNA if it's given a primer, which will provide a free 3' OH end, and these are usually 17 to 30, but optimally we use 20 nucleotides in length, single stranded DNA. And we use the forward and the reverse primers. That means you can call it a sense or antisense. And they are designed such that they flank the target region. So primers having the 3' OH group are required to initiate the synthesis, and they bind by complementary base pairing. So this is the template DNA. So after they get separated out, the primer, the forward and the reverse primer anneal to, so they flank the target region to be amplified, and then the extension step will amplify the or adds up the nucleotide. So other important components are obviously the DNA template, which provides the target region to be amplified. The DNTPs, which are the building blocks. So DATP, DCTP, DGTP, DTTP, and the buffer solutions for providing the optimal environment, the optimal pH, salt concentration, and which provides the stability of DNA polymerase. Some bivalent cations are also used, which acts as a cofactor, like the magnesium ion or manganese ions. The magnesium is most commonly used. Manganese can also be used for the mutagenesis because higher manganese concentration is associated with a higher error rate. So there are three basic steps: denaturation, annealing, and extension. So this is a template DNA, 94 to 98 degree denaturation. Then annealing at 55 to 70 degrees Celsius. The primers anneals its complement pair in the template. And then extension 68 to 72 degrees Celsius, where the polymerase binds to the 3' OH, free 3' OH, and then starts extending. And this is the first cycle. Usually we have two more flanking steps. One is initialization. The last one is the final extension. So there is actually an exponential increase. So from 1 to 2 to 4 to 8 to 16 to 32. So this means this is the exponential increase. So initialization is only required for polymerases that require the heat activation. So remember I've told you about the chemically modified or antibody modified polymerases. For that case, you need the initialization. Then it consists of 94 to 96 degree or 98 degree. if it is extremely thermostable polymerase. So then you need to use 1 to 10 minutes of the time. Then comes the actual denaturation at 94 degrees Celsius. Then comes the annealing, where the two target strands are allowed to cool in presence of the primers. So the primers will bind to the complementary pair present in the template DNA. So usually performed at 45 to 60 degrees Celsius for 20 to 40 seconds. So the TM, the temperature of melting, is a very important factor. So it is a typical annealing temperature we decide depending on the TM of the primer. So if the TM of the primer is, for example, 45 degrees Celsius, then its annealing temperature would be 3 to 5 degree below the TM of the primers. So the primer melting temperature is, the TM is a temperature at which half of the DNA duplex will dissociate to become the single stranded and indicates the duplex stability. So primers in the TM range of 52 to 58 degrees Celsius gives the best results. And the primers with TM above 65 degrees Celsius have a tendency for secondary annealing because of the more GC content. So the GC contents will give a fair indication of your TM. So it can be generally calculated by the formula TM is equal to 2 into A plus T plus 4 into G plus C. So number of A's, number of T's, number of C's, number of G's. So you will get to know the TM of that particular primer. So if you are using a forward and a reverse primer, they should have a more or less equal or comparable TMs, so that you can use a single annealing temperature for its annealing. Then comes the last step, which is the extension, which is performed at 72 degrees Celsius. And the time actually depends on the length of the target DNA to be amplified, as well as on the polymerase. So we all know that how much time it takes for the extension. So depending on that, you set the timing of your extension. And final extension sometimes may be required if at all some of the DNAs are left out to single stranded. So those can be double stranded or elongated, fully elongated during the final extension step. So the end result, we all know, can be done by running an agarose gel electrophoresis, and with the help of the DNA ladder, which is a molecular weight marker, we'll go and get to know the size of the band. So you have seen the how to run the agarose and all. So now let us see what all are the components of PCR in the reaction mix and their concentration. So DNA we choose 0.01 to zero point 0.1 microgram. Primers we use 20 picomoles. Then Fritz HCl, the buffer, we use 20 millimole at pH 8, magnesium chloride at 2 millimoles, KCl, and ammonium sulfate. Then DNTPs 50 millimicromole each, so in together 200 micromole. Then a thermostable DNA polymerase, so 2 units for each reaction. So volume should be around 20 to 100 microliter in the PCR tube, and the cycling condition could depend on the template and the TM of the primer. So you have 94 to 95 denaturation, 1 to 10 minutes. Then you can have 25 to 40 cycles. So more number of cycling is required for the less copy number of the template DNA. If you have sufficient copy number of the template DNA, then you can see a visible amplification in just 30 cycles. So denaturation, annealing, extension, and then the final extension. Now let us see the applications of PCR. So Number one is a selective DNA isolation. So the DNA, if you want to isolate it from the pool of DNA, then amplification and quantification of DNA. So this is mainly crucial for the forensic analysis from the criminal scene, crime scene. If you so only a trace amount of DNA is available as evidence, and also for the amplification of DNA from the ancient samples, you can use the PCR. Even for the medical application like this genetic disease testing, mutation testing, tissue typing for organ transplantation, diagnosis of cancer, that is in leukemia and lymphoma, even for the diagnosis of infectious disease like COVID-19, HIV, TB, spread of a disease in the disease organism in population, new virulent subtype, viral load, forensic application may you have the DNA fingerprinting. And there are several research applications as well, like the generating the hybridization probes for the southern and the northern blots, DNA sequencing, DNA cloning, sequence tagged sites. So the sequence tagged sites are something which are unique to the particular chromosome, just a single sequence, unique sequence of around 100 to 1,000 base pairs are present, which are unique to that chromosome. So those are called the sequence tag sites. And phylogenetic analysis of DNA also for the gene expression studies. So you convert the mRNA to cDNA and then see the quantitative via the real time or the quantitative PCR. You see the level of expression of the mRNA. There are some limitations of PCR as well, like the contamination, which is the most common problem. So usually if you have a particular lab working on the PCR particularly, so you should have a pre-PCR room, PCR room, and the post-PCR room. So you can find in the PCR room and the post PCR room, there are lots of uh, DNA in the environment. Either could be of the sample, mostly of the sample. Whereas in the pre PCR room, it is normally sterile, it is free of DNA. So the chances of contamination are far less if you set up a PCR in the pre PCR room, and then your PCR machine is kept in the PCR room, and then the post PCR room where you run the gel electrophoresis. So you then this way you can reduce the contamination. Then yes, one of the important limitation is that it can only amplify the known sequences. So for the novel sequence or the unknown sequence, if you don't know this sequence, how will you design the primers, the flanking primers? So it is one of the drawbacks. Then the next is primers can anneal non-specifically. So if you have some more or less similar sequences present uh, in the target DNA, which have the similar sequence, then it can bind non-specifically to that region and can amplify that region also. But that was not the the region of our interest. So you have to keep in mind while designing the primers that you should specifically design a primer in such a way that it won't co-amplify the unnecessary regions of the template. Then you can have some incorrect nucleotides incorporated during the PCR, which may give a false result if you sequence that. So incorrect nucleotides can be incorporated because it lacks the proofreading activity. Normally, we use tag DNA polymerase for the routine application, so it has got some error rate, but it won't change the whole sequence. But at a very low rate, you have the incorrect nucleotide incorporation. So there are several variants of the PCR. So in my next talk, I'll discuss you about these variants. So thank you guys. Get back to me if you have any query for all other previous lectures as well. We can have the online discussion via Zoom. Thank you.